So that's really exciting. Our AlphaFold3 just continues expanding on that idea. It's the newest iteration of AlphaFold, but it also models all these other types of molecules. So it's simultaneously predicting the placement of an ion, the conformation and placement of small molecules, proteins, and nucleic acids. So pretty exciting. We'll start with showing just some predictions. As you can see, pretty good. Like it's lining up with the native structure pretty well. It's predicting the nucleic acid, the small molecules, pretty much in the right area. Conformations may be a little off, but for the most part, pretty good. And these are some of the results for various different prediction types. So this first one of which is the ligands test. So this is how good the ligand is, like the conformation of the ligand when it's making that prediction, and this is like a success rate. Nucleic acids, also success rate is pretty good over Rosetta fold Na. And then it can also model covalent modifications. So this is like glycosylations or maybe a phosphorylation. And then it also models proteins pretty well. Some caveats here. These are success rates given 25 predictions. So yeah, take that with what you will. But the antibodies is with 5,000 predictions. So there's still a lot of room to be improved with antibodies because they're quite difficult to actually predict correctly. But this is, I think, exciting to see that we're able to model these things pretty decently. Let's continue. So very simply, it's quite similar to the original AlphaFold, where we have our inputs, we do some genetic database searches, and then from all these new MSAs and templates and stuff like that, we throw it through the model and get a prediction. But now we have the sequences for the DNA, the RNA, and the protein that we're interested in. We have which small molecules we're interested in, as well as covalent modifications, ions, etc. This is kind of just showing the calibration of the confidences. Once again, it's pretty calibrated. So the higher the LDDT typically corresponds to higher PLDDT, and this corresponds to all the different types of protein. So protein, the PTM score matches or is pretty correlated with .Q. For nucleic acids and proteins, the IPTM with ILDDT, and so on. So you can kind of trust the confidence metrics, which is just a nice validation to see. All right, let's break it open. So AlphaFold3, very similar looking to AlphaFold2. We take all of our inputs, throw it through these searches, and now we have the templates. We have our MSA for both nucleic acids, usually just RNA, and then proteins. And then we also, for the small molecules, generate some conformers because there's multiple different energetically favorable conformations of these small molecules. So we kind of generate a few of them to give the model some idea of what it can choose from. These go through the input embedder to kind of produce a few different representations. We have this green inputs representation, which we'll talk about later, and then the pair representation and the single representation. These represent similar things to what we saw earlier. However, there are some technical difficulties we need to talk about. So when we talked about AlphaFold2, each token in the transformer was a single residue. How do we really get that to work with all these other molecules? So for at least the nucleic acids, there are nice building blocks that we can choose from. So we can make each nucleotide its own token. So that's pretty straightforward. But for covalent modifications, for small molecules, it's a little more difficult to actually say what a nice token is for that. So for those, they actually decompose them into their own atoms. And so now each atom of these small molecules or covalent modifications now represent a token. So we've now broken a small molecule into multiple different atoms that represent a token. Our covalent modifications are also a bunch of atoms. So now this contains a bunch of tokens that are different types. So there's amino acid tokens, there's nucleotide tokens, there's atomic tokens as well. So that's kind of how they're tokenizing things. Now that's going to represent a single dimension of these representations. So now this is the number of tokens by number of channels. This is number of tokens by number of tokens by number of channels, and number of tokens by channels. Okay, did that make sense?
I have one question.
Yeah.
About conformer generation, is it creating a large sample? Is it creating representatives? How exhaustive is that?
That's a good question. I think it's just like representatives. I think they're using like RDKit to generate those, but yeah, I think that's what they're doing.
Okay.
Cool. Okay, so once we have these different representations, we throw it through some blocks. So they use 2 intermediate blocks to kind of take in all the information from the templates and the MSAs, which is maybe a little different from Rosetta or from AlphaFold2, where they had this explicit MSA representation that they carried throughout the EvoFormer. So a couple benefits of this is you no longer have to have this gigantic MSA representation in memory, because that was number of sequences by number of residues by number of channels. So it's this gigantic matrix. So we kind of eliminate that. We can kind of use these blocks to process all the information and squeeze all the information we can out of it. Same thing with the templates, although it's not that different from AlphaFold2, where they were able to squeeze everything out in the start. They've swapped the EvoFormer for a Pairformer. We'll talk about exactly what that means. Not terribly different, and it's quite similar to the folding block within ESMFold, as we'll see. And then they swapped the structure module for a diffusion module. Once again, this kind of just takes all the information that was processed at the beginning and starts transforming it into a concrete structure. We'll talk about exactly what this means with the diffusion stuff, but then once again, you have your final output structure with its confidence as well. So this is what the MSA module looks like. If you squint your eyes, it looks like the EvoFormer. And there's a good reason for that, because it really just takes the MSA representation and tries to extract all of the meaningful covariation using primarily these transitions and this outer product mean. It extracts all these covariations to kind of get that structural hypothesis, and it starts baking it into the pair representation. The pair representation is also being updated using these triangle updates, as we've talked about. So there's the triangle multiplications, there's the triangle attention, and then there's that whole transition where each residue just kind of updates itself based on different properties of it. And then we have our final pair representation. So all these get updated, and we just have a pair representation. No more we don't have an MSA representation coming out of this. And as I mentioned, it looks very similar to the EvoFormer. We have very similar operations. We have these operations are replacing the column-wise and pairwise attention, but then we still have all these triangle updates. The Pairformer now does very similar things. It does the triangle attention, the triangle multiplication to update the pair representation even more. But now we also use our single representation with just single attention, and we bias it with the pair representation. So now the pair representation is giving information about its structural hypothesis to the single representation, which is able to update itself and learn about exactly where maybe things need to be. And then these also get fed back in a feedback loop. So there's 48 blocks of these, so they get processed and really extract all that structural information and kind of try to place it all in this single representation. As I mentioned, this is quite similar to the EvoFormer and the folding block. It's honestly almost identical to the folding block. Maybe the operations are a little in a different order, but it's pretty much the exact same. So they're really kind of just repurposing some of these ideas and just maybe like reframing it. So as I said, they replaced the structure module with a diffusion module. We haven't really talked about diffusion yet so far in this bootcamp, so I wanted to share a slide that kind of like tries to illustrate the idea of diffusion. So the main idea is that we have a bunch of data. So in our case, we have a bunch of native protein structures with its other interactions, other molecules as well. And they iteratively corrupt that data with noise. You just add noise to it. And this, eventually, if you add enough noise, you're getting something that's just completely random. We have this completely static picture in this case. This is the forward diffusion process, or the noising process. Once we have this process, we have multiple steps of increasingly noisy images, or proteins, or structures. And then we have a model that is trained to reverse that process. So it's learning to iteratively denoise each noisy image into the previous step. So we have these increasingly noisy images, And now we take the noisiest and we try to remove a little bit. We try to take a step back. We keep going and going and going. And eventually, if the model was trained well enough, we'll get a sample that looks like it came from the data distribution. So it looks like it came from the PDB in our case. And yeah, only the reverse diffusion process is learned. We have a model that actually learns each individual step. Any questions on diffusion?
So there might be a naive question. Why would the forward diffusion not cancel out the reverse diffusion?
Cancel out?
I mean, like when you do a forward diffusion, you have a static image. And when you do the reverse image, you get the original image back. So in a way, you don't actually gain anything.
So you're right. So when you kind of do the step backwards, you are training the model to reproduce the denoised, the ground truth denoised version of it. But when you start using it in practice, you're starting to give it these binoised image. So when you're generating a new sample from the model, you give it complete noise. It's completely random. It probably has never seen this noise confirmation before because there's so many different possible noises. And then it will learn to denoise that. And because it's learned to do it well, it's learned to reproduce the noised ground truth samples. It should produce something that is similar enough to the data distribution.
I see. So the input is the noise.
When you're doing inference. This is only really, the forward diffusion process is only really relevant for training because we only have data examples. We have a bunch of data examples and then we can corrupt them with noise. And then we learn that reverse process.
Like the noise we're adding, like in the forward noising process, is random, right?
Mm-hmm.
So what I'm wondering is, for the same picture, we should theoretically we can get many different noises in the end.
Yep.
But I'm wondering if the image that is, if we took for the same image, we do the forward noising, we created the fully noised image and denoise it. Do we actually get the same image, or do we get a similar image, or can we get a completely different image?
You'll likely get a similar image. You probably won't get the ground truth, but you're training it to try to reproduce the ground truth. So you're exactly right. When you noise this, it's also not like it's a stochastic process. It's not deterministic. So even as we're noising it, we're adding random noise to it. So even the forward process for a single data example will be different every time you noise it. So the model has to kind of learn to adapt and be able to, given any kind of noisy version of it, try to make it more realistic. So yeah, it's a very complex task, but it eventually learns how to do it quite well.
If two images are close are similar, let's say in meaning in our regular space, regular images, do we assume that the noise images created from those images are also somehow similar? And you could take these completely nice images and cluster them, and you would actually get some meaningful information.
I see what you mean.
Is some kind of embedding, like?
Yeah, so that's a very interesting question, and kinda, yes. It's kind of a difficult question to answer without getting too technical, but when you're getting to, so one thing you assume is you have two distributions at the end, either ends of this process. You have your data distribution, you have all of your ground truth PDB structures of your proteins and other molecules. At the other end, you have a prior distribution. In our case, completely just uniform Gaussian noise. In this case, it's Gaussian noise in 2D. In our case, it's Gaussian noise in 3D. And the idea is we can map trajectories from that noise back into the data distribution. But because this is a very stochastic process, you're actually learning a SDE, a stochastic differential equation, because you're learning that it's actually not like a straightforward path. So while the images that are like similar to each other in the data distribution are being noised, they start to lose their semantic meaning and start to kind of become quite similar to every other thing because you're starting to add more and more random noise. So early in the process, similar proteins will look similar to other similar proteins. but that'll quickly change. Did that answer your question? There are other ways. So if you're training a diffusion model, I don't think you'll have a nice clustered representation of the noise distribution at the end. But if you're using a variation of it, such as like flow matching, which is a different way to do it, but it's a straighter path. And so in that case, your prior distribution is actually more clustered than in a diffusion stochastic differential equation based method.
Okay, but in training, you actually expect the model, the image creating with the reverse diffusion to be the same as the original image, right?
Yeah.
So theoretically, it should contain some information about the original image. It shouldn't be purely noise, right? Because you could.
It does, right. It has some information. But if you like take, so typically you define a number of time steps for this process, maybe 100. If you take 99 steps, you're so close to noise that pretty much all the signal you have of the original protein is gone. It's pretty much just noise at that point. So it's really hard to identify what the ground truth structure is. So you take a small step that maybe makes it look more protein-esque. You keep taking these small steps, and usually for these models, around 60% of the, usually around, how do I phrase this? So if the diffusion time step is from like 100 to 0, so 100 is complete noise, 0 is complete unnoise, so the data distribution. Usually around time step 60, or around that era, 60, 50, 60, you start to really converge to a very specific structure. So the model starts like really converging to a very specific structure of that protein, and then the rest of the steps, the last 60 steps, are usually refining that structure. So figuring out the more finer details, but at that point you kind of have a rough image of what it should look like. Like we know at this point that there's a nice mountains. We just need to kind of fill in the details. So that's the last little bit, is it just kind of resolving the fine atomic details.
So transformation of like I+1 noised image to I noised image is the same like I+2 noised image to I+1 noised image?
No. So there is also a noise schedule that depends on what time step you have. So usually you you add a little bit of noise when you're very close to your data distribution. So when you have a really low time step, you're adding a very little bit amount of noise. So you should be making very minimal changes to the structure. But then you start rapidly increasing the amount of noise you're adding. And so by the end, you have just like a dramatic shift, and you have pretty much just noise.
But what about the denoising steps? Are they the same?
So the denoising, it'll learn to reverse that process. So like whatever noise schedule you're teaching it, so you kind of have to assign a noise schedule a priori. So like you have to decide what noise schedule you're going to use when you train something like this. But then the model should learn to reverse that specific noise schedule. So if you change the noise schedule, it might like change kind of the outputs you're getting from the model, because it kind of also affects the reverse diffusion process. You're asking some interesting questions, but it's hard to like answer them without getting too technical. But I'm happy to talk more about that later if you want.
Okay, yeah, I will stop now.
Any other questions?
So if you have two noise distributions that are exactly the same, will you get the same starting image or data distribution?
So it's a stochastic process, so no. During each denoising step, you're also adding a little bit of noise. So this constantly changes the training dynamic. So like you'll bounce around while you're denoising, which gives it more diversity, but also may, like, I don't know, bump you into a different final place. Cool. Okay, so I wanted to show a little bit of image of what this may look like in AlphaFold3. It's hard to actually say what it does look like because we don't know. We don't have a nice video of that, but this is AlphaProteo, which is based on AlphaFold3, but is from Isomorphic Labs. They have a design protocol using AlphaProteo, which I think is probably close enough to AlphaFold3 to show that. So in this case, they're designing a binder towards a target protein. And let's start over. You can see that it's denoising. So it starts complete random noise, all located very similar to each other, and then it rapidly starts to figure out what the global structure is, and then it starts to refine the inner details. So one thing you'll notice is that it rapidly starts to figure out what the overall global topology is. So it's pretty much decided at this point. You know, we have a certain amount of beta strands, we have a single alpha helix, and now it's trying to resolve the fine details of the atomic placement of the side chains and all the other components of it. So that's kind of what the diffusion process looks like.
I have a question.
Yeah.
You mentioned yesterday with AlphaFold2 that we usually start all of the coordinates at a single location and then expand outward. Is that the similar?
So they're starting from random noise. So it's a sample from the Gaussian distribution.
Right.
So they're not all at the same location.
Oh yeah, sorry, I don't mean the same location. I guess just a condensed location.
Yes.
I remember
Yes.
Ben mentioning as well that instead of having it all at a condensed location, you could actually have it start similar, like a template, for example.
Yeah, so they typically just start from a standard Gaussian distribution.
And then that just is defined by a certain angstrom sphere, I'm guessing, or?
Yeah, pretty much.
So the receptor protein in that video, is it kept rigid?
In this case, it is, yes.
So in this case, how does it know to bind the
It's hard to exactly say because they didn't release like a lot of the methods for AlphaProteo, but I assume that there's some sort of like epitope information that it's being biased towards. And you can kind of see that already by the placement of the initial cloud of the noised coordinates at the start. So I think it's something like that. With RF diffusion, we can specifically say, hey, these are the epitopes we want you to target. Go move it over there and start binding that. So I assume it's something like that. It's hard to exactly say though. So this is just a comparison of what the diffusion module looks like with respect to the structure module. So it's kind of similar in the sense that we have our pairwise information, our single representation information, as well as our kind of noisy current location of where the positions are. You can compare this to the IPA structure module over here. So we have our random, maybe not random, but starting locations for our backbone. We do some attention that is very local on each specific atom, and they just do a bunch of attention really to try to incorporate all this geometric information and really combine these different representations into something that they can make an update step for each of these individual atoms in the process. So at this point, we've kind of broken down the token representation, and we're now diffusing each individual atom.
The structure module is deterministic, right?
The structure module itself is, the diffusion module is not. Yep.
That's a big difference between AlphaFold2 and 3.
So when you run AlphaFold2 multiple times, you still likely will get different answers. That's probably because of the subsampling of the MSA at the beginning. But with these, you'll get a different answer every time. Yeah. Did what I say about the decomposing the tokens into individual atoms make sense? I can try to explain that more.
I would love more detail.
Okay, so I said each token in like the protein space represents a single amino acid. In the nucleic acid space, it's a nucleotide. Small molecule, each individual atom is its own token. At this step, we do know which atoms correspond to each token. So like we know what atoms are inside of a arginine. At that point, we have that token and we kind of connect it to each of its atoms, and we're diffusing the atoms. So the atoms are moving around, and then eventually they'll talk to the token. The token will send information back to the atoms. So that's kind of what's happening. And all the diffusion is happening on each individual atom, but they're kind of being guided by that token's information.
So each token have different number of atoms.
Yes.
I'm guessing they have other things like distances for the connectivity within that. No.
No. It learns how to do that. Somewhere in here, they give it what an idealized version of each of these tokens looks like. So they have like an idealized version of phenylalanine. And that's to help guide the model about like what the bond geometry should kind of look like. But it is completely unconstrained in the diffusion process, which is kind of crazy. Yeah.
So I guess like with how this thing does the tokenization, is it able to like handle like say non-canonical amino acids well then?
Yes. So it handles covalent modifications, but also non-canonicals. Yeah.
So we talked about how like mutations, like the model, like AlphaFold and all these other models don't like handle them particularly well. Does this model, is it better about handling like post-translational modifications, which we know have like structural importance, or is it kind of not?
So it is much better at predicting like the location of the post-translational modifications, but I think it still has the same limitations with mutations. I don't think.
Is that because of the MSA?
I think it's primarily because the MSA. Although, because we're no longer carrying this MSA representation throughout the model, it has less of an impact in AlphaFold3 than AlphaFold2, but it's still quite an important aspect of it.
So.
Okay. Oh, another question?
Could I ask for about, but actually not about this, but the earlier stage. I still don't really understand the embedding of the small molecules and what happens with them in the model.
Yeah, so the small molecules are represented as individual atoms within the structure itself, like as a token, right? Each atom is a token. But it is informed by like the conformer generation, so it knows which atoms it should be connected to, and it kind of knows what the general shape of the small molecule should look like, because we kind of give it ahead of time what the conformers' possible arrangements of those atoms are. So it kind of already knows, like, I'm connected to these guys, and I should generally have one of these many shapes.
For example, where are they in the pair representation? Because we see the input embedder creates this pair representation.
Yeah. So the pair representation is number of tokens by number of tokens. Yeah. So it's somewhere in there. Like each individual atom is its own token, and it has an interaction with every other token in that pair representation.
Also here the pair representation isn't like first, second, third.
It's first, second, third token, which when you're doing like a protein and a small molecule, the first part of that will be residue-residue interactions. And then when you start getting down here in these portions of the matrix, you have small molecule atom residue interactions. And then if you're in the small corner down here, this is small molecule atom interactions.
Thank you.
Yeah.
Oh, I guess question about like the noise on the diffusion.
Mm-hmm.
Is it like adding noise to both like the query sequences that you have and like the structures that it's pulling from the PDB, or is it doing it to just like one or the other?
So at this point, we have just random noise that we've sampled. And we've also tried to extract all the possible information we can from the templates and the MSA. So now those are no longer being noised. We kind of just throw those away. We don't regard those anymore because we've extracted everything we want from them. And the noise is random at first, and then it starts to become less random and more structured as the diffusion process comes. The losses are quite similar to AlphaFold2. However, we kind of swap some of them. For example, we swap the FAPE loss for this diffusion-based loss, so it's really just trying to recapitulate the ideal denoising step. It's essentially what the diffusion loss is doing. It has a similar histogram loss on the pair representation, and then some confidence losses just to make sure that it's still producing something that is, the model knows how much error is in it can estimate that error. Some interesting points, because we are doing this diffusion loss on each individual atom, we are no longer constraining the bond lengths and angles. We're also no longer strictly enforcing chirality, because the FAPE loss enforced chirality, this no longer does. But also because we're using each individual atom, this process needs to be equivariant. And the way they kind of do that is they don't do anything special with the architecture. They actually don't do anything with the architecture. They just kind of use a standard neural network components for that. But they augment their data. So you have a protein. Like I said, if you rotate it, translate it, it's the same protein. So they just randomly augment their protein. They rotate it and translate it in space for every diffusion step, pretty much. And that teaches the model how to obey equivariance without explicitly baking that into the architecture itself. So that has sparked a lot of controversy, like whether equivariance is even necessary to bake into the architecture itself. This is still a really unanswered question, and there's a lot of people talking about that now. So I think it's a very interesting time, at least for that aspect of things. Want to talk? Oh yeah.
I guess with the like equivariance thing, have there been any like extensions on AlphaFold3 that have like tested like what happens when you knock out that loss function, or?
Unfortunately not, just because it's so new and everything. It's still a closed source. So we'll talk about it, but there are people trying to replicate results from AlphaFold3, but yeah, it's still to be determined at this point.
Okay.
It's too recent, yeah. Okay, so I want to talk about some limitations. These have different limitations than AlphaFold2, although some of them are similar. The first of which is it needs a lot of seeds for difficult targets. Like I said, for antibodies, they used 5,000 seeds in order to get that 60% success rate. And here they show these plots of, as they increase the number of seeds, how does the percentage of successful docks change? So this first one is just successful docks. This one is high accuracy docks. So you can see it's much harder to get a high accuracy correct location. But increasing the number of seeds has a lot more dramatic effect for AlphaFold3 than AlphaFold2, which is interesting probably because of the diffusion process, but also by ranking these predictions using the internal confidence metrics of AlphaFold3, you can still identify these nice quality predictions. So it's kind of pointing to the fact that the confidence parts of the model know what it wants. They know what a good structure looks like, but getting those structures, that global sampling of those structures is really difficult, and it takes a lot of tries in order to get that global confirmation correct. Yeah.
Oh, what do you mean by increasing seed? To do the training from multiple times, or to do the inference from multiple times?
Inference multiple times. So you change the random seed, and you'll change some of the stochastic process of the inference itself.
So I only get the results with the best PLDPT or something? Or
Yeah,
inverse them?
it's something like that. They don't exactly use just PLDDT, but they combine it with like IPTM and stuff like that. Cool. So that's a major limitation. Like I said, it also doesn't enforce chirality. So around 4 to 5% of the like ligand, the postbusters, in the postbusters data set, they have around 4 to 5% of structures with wrong chirality. So here we have this small molecule that has the wrong chirality. So this is a major issue. You might want to make sure to check the chirality of your small molecules if that is what you're predicting. It's also prone to hallucinations, which is just kind of a problem with diffusion models themselves, because they're trying to make something that looks very data-esque. For parts that it has no idea what to do, it'll still try to force it to look like a protein. So this is an example of a nuclear pore protein where only this very small region is resolved. The rest of it, like 1,800 residues or so, are unresolved. AlphaFold2 multimer would have no idea what to do with that. It is deterministic, at least in the structure module part, and so it just kind of makes spaghetti with it. So you can't really trust it, but it makes it very clear that you can't trust it. It's also very low confidence, as shown by the orangish red color. So yeah, you can kind of see what AlphaFold2 multimer is doing. AlphaFold3, however, will make it look very structured, which is very misleading, because like if you look at this just at first, you're like, oh, this is very structured protein, but in reality it's not. And it knows it's not. It's showing the confidence is really bad. So it knows it's not real. You can't really trust these regions, but it's still making them look protein-like. So one thing they actually did was fine-tuned AlphaFold3 on a bunch of AlphaFold2 multimer structures. So they tried to teach AlphaFold3 to reproduce a spaghetti-like structure on regions that it's really uncomfortable about. So yeah, I thought that was a fun thing to talk about. And yeah, any questions on that?
So is this after fine-tuning that it still does that?
Occasionally it still will, but it is much better at producing spaghetti.
Wait, so you actually wanted to produce spaghetti? That's not to be serious?
Yeah, I mean, if you just take a look at this, you get a very wrong idea of what the structure of the protein looks like. However, for this, it's a lot more obvious that it's uncomfortable with these like long regions that has no idea what the structure is. AlphaFold3 is still not happy with it because it's very confident about it, but the output is very misleading. And so it's kind of just like...
So are there the chain breaks like in the AlphaFold multimer model, or is just the image?
I think it might be chain break, yeah. I think that's an issue that a lot of models have with these large proteins. Like even RosettaFold2 has issues with that. Like they do form chain breaks with just, it's a major issue with large proteins. And that's something RosettaFold2 really had to deal with in development, if you listen to some of Frank's talks.
So in this case, the ground truth is an experimental structure or AlphaFold?
The ground truth is this little nuclear pore region that was resolved. The rest of it's unresolved.
Oh, the rest of it.
Yeah. So it's still like making a prediction for that unresolved region. However, it's only really being trained to predict this correctly because that's all we know that's right.
Was the diffusion trained with intrinsically sorted proteins? I mean, how does noise affect an intrinsically sorted protein?
I mean, the noising process is the same for intrinsically disordered proteins or regular proteins. It's just the model tries to form structures that aren't there. Yeah, I don't know if that answered your question, but.
No, my question is if I give a noise of an intrinsically disordered protein and I try to denoise it, will it make structures?
If you use the version before fine-tuning, yes. If you use it after fine-tuning, probably not, but maybe.
I don't understand really the intuition of making spaghetti. Like, yeah, the confidence will give you already the information of what to trust and what not to trust. But if you're teaching it to generate spaghetti, you're taking some of the physics out of the sequence itself, right? The sequence of those regions that are folded as alpha helices, maybe it's because they are alpha helices, but they are not in the right relative position to everything else, right?
Well, I mean, the model's not even sure that these are alpha helices.
Yeah, right.
And like, it was trained on just a small region, so it literally has no idea what these should be, but it's still forming structures. It's forming alpha helices probably because that's the simplest structure it can form. It's super easy to make alpha helices with these diffusion models.
But the alpha helices are more physical than a spaghetti.
That's true, but it's very misleading. Because now you'll have the idea that these are alpha helices when they really, you have no idea what they should be.
Right, but the color will tell you that.
But you have to look at the color.
Exactly. But if you give it a spaghetti, then you're, let's say, making the model worse because you're like teaching it to generate something that is not fit.
That is a problem too. So like, yeah, whether this is the best tactic to combat the hallucinations of these like structured regions when they shouldn't be structured, it's hard to say whether that's the best tactic, but that's what they went for. Yeah.
Out of curiosity, if you took like that structure from the AlphaFold3 and put it into the modeling, like in Rosetta, just run in Rosetta Monte Carlo simulation, would you expect those low certainty regions to be more flexible, or?
I feel like it'll probably not change the structure too much, just because I feel like Rosetta will probably try to keep it quite similar to what it was already given, because like these will still have that nice hydrogen bonding pattern that is canonical with alpha helices and stuff like that. So it's probably not going to want to change that because it looks nice energetically. Like these are very nice structured regions that it's predicting. They're not right because we don't know what it actually looks like. But I think Rosetta will interpret it as like a nice structured region and it probably won't change that much. Yeah.
Do both methods go to amber relax?
I don't remember if they do amber relax on these. I think so.
AlphaFold2 structure. To me, it looks like there's some clashes.
Yeah. Yeah. AlphaFold2 still produces clashes. AlphaFold3 still produces clashes. That actually leads me, I actually missed out on that limitation. But so sometimes they have seen a lot, so this does occur it's not uncommon to see chains that are completely overlapping with other chains. And that's mainly because the diffusion loss doesn't incorporate any like physics information. It's just trying to recapitulate a protein-esque shape, but it doesn't necessarily know that these should not be overlapping. It wasn't really taught that. So it still will produce a lot clashes of sometimes just chains literally on top of each other. They're very structured, but they're very just literally on top of each other. So that is an issue. And it's also common with like nucleic acids. I think that's one of the issues with nucleic acids too. Another one of the limitations is it will produce multiple states because it is a diffusion process. It can sample multiple conformations. However, the existence and the occupancy of these states is still isn't guaranteed at all. So this is an example of a protein that has two different conformations, an open and closed conformation. When binding a small molecule, it transforms into this closed conformation, this hollow structure. So when AlphaFold predicts it without the small molecule, it's closed no matter what. When it's produced with the small molecule, it's still closed. So it's not predicting the correct apo structure even without the small molecule. Whereas we know that it should be these regions should be a lot farther apart when there is no small molecule. So it's still not perfect about sampling multiple states, so still be careful with that. And the final limitation I had is still server only. If they keep their word, then we'll have the open source version sometime in November, but that's to be seen. So hopefully, I'm hopeful, but we shall see. As I mentioned, there are some people trying to replicate it. So HelixFold3 is one of the replication by Baidu in China. There's also Kai1, which also has some extra like knobs to play around with. For example, you can include experimental data in some of its predictions to maybe bias it towards something more realistic. It's also, Kai also kind of boasts that its single sequence prediction is much better than like AlphaFold3 single sequence prediction. And then I also want to shout out Lucid Reigns, who's doing a really Herculean effort to try to reproduce AlphaFold3 in complete PyTorch. There's a lot of people contributing to that project too, so it's really cool to see how that's progressing, but it's still pretty early.